Thank you, Andre, for this lovely introduction. Thank you for the organizers, for the Danone Institute for inviting me. So I'm talking about dietary modulation, and I have disclosures today because I am a co-founder of Symbiotic Health, and I will show some of a few slides of their probiotic work, so I would like to declare this. So I wasn't aware that I was working on aging, so I was very surprised I got invited, but I'm definitely interested in how the microbiome interacts with these chronic diseases that have already been mentioned several times, and how this is then shaped by diet. And since many of these chronic diseases are linked to older age, and giving also the fact that we should be probably eating healthy all through our life to prevent these, I probably have been working on healthy aging all around and all along. So this is why I then really enjoyed actually presenting this presentation. I'm also surrounded in Cork by absolute experts. So this is Paul O'Toole, a grumpy Irishman. And he is one of the world leaders in this area of microbiome and aging, has published very, very important papers showing that we have a dysbiosis, for example, the older we get, especially in not so healthy aging, where we have pathobionts going up, protective taxa going down. We also see inflammation, or how some scientists then refer to this as inflamed aging going up, often related to a disruption in barrier function. And then this is linked then to other, readouts of aging, such as frailty and other, cognitive struggles. Yeah. This is one of the classic papers. This is a slide Marcus Claesen provided me, who is also a professor in Cork. This is their famous paper on the elderly, published in Nature in 2012. Really interesting work, showing one of the first studies showing how diversity actually declines in older age. And very, very interesting then ecological findings. For example, if you plot their fecal microbiomes, you can see then the elderly actually cluster pretty much with younger controls as long as they still live in the community. And when they then move more into care homes, their microbiome really degrades. It goes down in diversity. Obviously, there's a lot of ecological factors that then contributes to this, but they also already did preliminary analysis linking this then to the unhealthier diet that is served in these Irish care homes. Yeah. And that obviously then already, made them suggest, there is a huge, rationale to modulate with diet the microbiome to support healthier aging. And this is my outline today, so I'm going to show you some evidence that the microbiome is in fact linked to longevity and aging. And because it's very hard to establish cause and effect from human studies, I'm going to show you some evidence from rodent models. I'm going to show you some evidence from the nutrition field, how eating is linked to healthy eating. Then I'm going to show you two human studies that are, I thought, intervention studies on diet that I thought are of relevance as it relates to aging. Show you some of the microbial metabolic mediators that are likely to link diet to healthy aging. And finally, look then beyond, diet and for other microbiome-centered treatments and a potential role for bifidobacteria. So one of the few advantages of advanced age is that you know the older literature, and this is one of the very, very, and there is a lot of fascinating work on animal models as it relates to aging. So this is a publication from 1966, looking at comparing germ-free versus conventional mice, and very much surprisingly, these are the mice with a microbiota, and here you can see the germ-free counterparts, and as you can see, there is a substantial elongation of lifespan in germ-free mice, and the same work has been replicated also in rats. Also, at least in this dietary context here, the microbiome actually shortens lifespan. A study then later on confirmed this finding here, that the germ-frees actually live longer, but what it also showed is that this is on an adequate diet. If you dietary restrict these mice, then actually the negative effect of the microbiome goes away and even becomes slightly beneficial. And you can also say dietary restriction is even more important than actually the presence of a microbiome. But there's definitely an interaction between diet and the microbiome as it relates to lifespan. And then more recently, some really nice mechanistic work that was published in the last 10 years. These are 2 studies using mice and causal experiments in which actually older microbiomes are transferred to germ-free mice or through co-housing. These experiments actually confirmed much of this older literature that the microbiome is really often costly and that germ-free counterparts live often longer. They established causality. The microbiome is really causally related. Very elegant work here with transfers or co-housing. And finally, they define some of the mechanisms that I've already introduced, that you have a dysbiosis because older microbiomes can be transferred. This is then linked to reduced barrier integrity. You have more bacterial translocation, and then you have this systemic inflammation that is then linked, to aging phenotypes. So very, very interesting, causal, foundations of this exact field. And I also think this raises some, evolutionary questions about, how is the microbiome actually, what is our relationship with the microbiome as it relates to aging? Because obviously the microbiome was important in evolution. We have clear contribution to fitness, but this is obviously during reproductive age, and during as we get older, this seems from these animal models that the microbiome might become costly, and there are some detrimental effects due to barrier decline, bacterial translocation, and then inflammations. And this could, of course, now just be an evolutionary trade-off, meaning the microbiome has evolved to support fitness during reproductive years, but it also, in evolution, it could also be antagonistic pleiotrophy, meaning that it actually evolved to shorten lifespan, and by this actually increase the fitness of the population. And the first time I read this in a publication was here in a paper authored by Ruth Ley, who I'm really, really pleased to be on the podium. And this was really just one of the many times where a publication of Ruth completely blew my mind. So the microbiome might actually have evolved to kill us off when we are getting older to then, generate space, for the younger offspring. That's a hypothesis, of course. No matter if that's true or if it's just a trade-off, it's definitely that there can be a cost associated, and these animal experiments showed this is diet dependent and again provides a rationale to look at diet. So how should we eat to age healthy? So this is a very recent publication in Nature Medicine by the folks from Harvard School of Public Health. They looked at different, at their big nurses studies and health studies, so hundreds of thousands, looks how diet is linked. And the best indice actually that links with different, hallmark of aging, healthy aging, chronic diseases, survival past 70 years, cognitive health, you can see is this alternative healthy eating index, performs even better than some indices around Mediterranean diet, performs much better than actually a healthy plant diet indice, probably because there's a lot of not so good plant, processed foods. The reason why this is probably working so well is it's an US population, so it looks at fruits, whole grains, nuts, legumes, but it also specifically looks as detrimental, to processed food, sugary drink, but also then processed meats, where there is very, very strong rationale that they interact actually with the microbiome. If you look at the individual foods, you can see the usual suspects here of all the beneficial fruit, whole grains, vegetables. Yogurt shows up very high up, so the Danone folks didn't tell me to mention this, but it is very good, and it also has a FDA claim on the prevention of type 2 diabetes. That's important, but what is more important is wine shows up here. So you can really elongate your life by moderate consumption of wine, unfortunately. And then what is probably just as important is all the negative stuff here, and red and processed meat show up, trans fats, so also the usual suspects. But again, in terms of the microbiome, this is probably very important. There's a second study that I thought was very interesting in a UK cohort, also showing that you can actually extend your life expectancy by 10 years. Again, the usual suspects, you can see very, very high consistency. And what is very interesting in these studies, in the models, is that they show you can extend your life expectancy when you start eating healthy in your 40s, and if you start eating healthy in your 70s, it's only around 4 to 5 years that you can add. Again, showing that you should probably start eating healthier a bit earlier on. But there's still, no matter when you start, you can add life expectancy on average. Yeah. So how does this relate to the microbiome? This is a paper we published, a review actually years earlier, but I can tell you there is not a single disagreement with these studies on what is proposed. You have the usual suspects here of the healthy foods, and probably very little surprise: fiber, short-chain fatty acid, phytochemicals, polyphenols. But then what is probably just as important are these negative aspects: the processed foods, the saturated fatty acids, and the then metabolic, problems that arise from consuming, for example, high amounts of saturated fat, and I'm coming back to this in terms of bile acids, but also a higher proportion of protein, which is then fermented in the gut microbiome to detrimental outcomes. And again, high protein diets, again, in mouse models are actually linked to shorter life expectancy. So what do we know from intervention trials? This is, again, a study led by Paul O'Toole. This is, stands out. This is the New Age study. It stands out because it's a large intervention trial with a Mediterranean diet in 612 adults, several countries. They put them on 12 months with dietary advice, showing very nice that first, markers of inflammation and chronic disease and frailty go down. This is then linked to bacteria that are correlated with frailty and cognition. You see increases in short-chain fatty acids, reduction in secondary bile acids, also a reduction in fermentation output from proteolytic fermentation. This is independent of age, interestingly, but there's also some real issues with actually having the elderly stick to a diet like this. So there are definitely challenges around this detention here. Some of them don't have teeth anymore, and also some of them just don't really like the diet as much. Yeah. Some of the data here, you can see we had a lot of talk at this conference about Faecalibacterium prausnitzii, Roseburia, other short-chain fatty acid producers. In here, for example, we have certain Collinsella, but we also have Ruminococcus torques, a known mucus degrader. We have then strong correlations which very much make sense with marker of inflammation, and then reverse or positive or negative dependent on which direction the bacteria go, but then also correlations which actually markers of aging, such as hand grip strength, for example, and that is pretty consistent amongst the different countries in which this diet has been performed. So very nice. Yeah. So I would like to show you some of our own study. So this is the study on the non-industrialized type diet that we published in Cell last year. This is not a study in an aging population. Actually, the average age was 25, but as I mentioned, should probably start eating healthy early on. But it's also relevant because it was in contrast to the study we've just mentioned, and Aaron mentioned that it's hard to control for diet. So this is a strictly controlled diet study in which we prepared the meals and actually gave them. And it's also interesting because it's a diet that mimics the dietary pattern in non-industrialized populations, which show often very low levels of these chronic age-related diseases. So this is a fascinating paper in Lancet showing that a tribe in Bolivia, the Siame, have very, very low levels of heart disease, very low level of plaque in their arteries. This is a publication from rural Papua New Guinea in a population with very little stroke and heart disease. And then again, in the Sami, there's evidence that dementia is actually very low. So it is worth to considering how these populations eat. This is not primarily driven by life expectancy, because life expectancy is short, and this is driven by infections, but if these individuals go older, they still have much, much lower levels of these chronic diseases. This is, again, this Lancet paper showing that there's substantially less plaque in this Bolivian tribe, 5 times actually low, again, and then resulting in much, much lower levels of actually heart disease. So how do they eat? Actually, it's not an extreme diet, but it is actually quite high in complex carbohydrates, low in protein, very low in saturated fat, and then very characteristic of these non-industrialized populations, very, very high in dietary fiber, more than we actually recommend in dietary guidelines. Of course, no industrialized foods and no added sugar. And so this is very similar to this non-industrialized type diet that we developed that was based on the eating patterns in rural Papua New Guinea. Lots of shout out goes to Anissa Ahmed, who is now a postdoc in the University of Alberta, who turned this dietary information into meals that are actually acceptable to Canadians to eat and to be tested in a human nutritional trial. So this is the characteristics, very much in line with how these Sami eat, but also in line with what has been linked to healthy aging, very high in dietary fiber, for example. And then thanks to a collaboration with Catherine Field and Carla Brado at the University of Alberta, we could do a strictly controlled nutrition trial where every single meal was handed to the participants. And poor Anissa, during her PhD, had to spend 1 year in a kitchen cooking meals for inhabitants and handing them actually out. Good thing is, it really shows very strong findings then. So these are the microbiome shift. Strongest is the genus Bifidobacterium, but then we see Faecalibacterium, Roseburia, similar to what Paul O'Toole saw with the Mediterranean diet. Bacteroides, for example, goes down. Bilophila, a pro-inflammatory bacterium, goes down. And here again, Collinsella going down. So very consistent findings. If you look functionally, the fecal pH drops. And for me, this is exciting because I think this is a really underappreciated marker, the importance of actually the pH in the gut. And I was involved in this nice review here by Tine Rask-Gleit, showing how many metabolic processes you can actually change by reducing the pH. One being, for example, the reduction of proteolytic fermentation, which we in fact showed in a reduction of branch-chain fatty acids. We also saw an increase of this metabolite indole-3-propionic acid, so that was in plasma. It's a microbial metabolite linked to a whole range of benefits. It's neuroprotective, for example. And then we also saw a reduction of secondary bile acids. And there have been already a few talks around secondary bile acids, and they're important in signaling. And I don't want to take any of this away, but these are also established carcinogens. And there's just a really nice paper by Tom Clavel and other individuals clearly causally showing that secondary bile acids are actually causing colon cancer. So I do think that this is, and again, also this is consistent with what Paul O'Toole showed in the Mediterranean diet study. Yeah. What happens to cardiometabolic risk markers? First, these individuals lost weight despite the fact that this was a calorie-controlled study. So you actually consume or less accessible calories at least. And you can see very, very strong effects. Fasting glucose goes down by 6%. These are healthy individuals, but LDL cholesterol goes down 17% in just 3 weeks when you go onto this diet, and CRP, mark of inflammation and heart disease, goes down 14%. So very, very strong effects on cardiometabolic risk markers, and we work with machine learning gurus like Dan Knight and Russ Kreiner showing that. And again, this is not establishing causality, but if you look into the metagenomics, actually one predictor or the dominant predictor of the reduction in CRP are actually mucin targeted to total metagenomic reads, at least suggesting that actually an improvement in barrier integrity might be responsible for this, anti-inflammatory effect. Yeah. So this, again, this wasn't a study on the elderly, but it probably still shows certain mechanisms by which we can modulate the microbiome to improve then healthy agings. And, I'm sure Sean is probably going to talk a little bit about this too. So some of the usual suspects, butyrate, for example, but then also I mentioned mucus degradation and also, the improvement of this barrier function and then anti-inflammatory effects. And obviously, this is complex diets. They're very reliable. We actually see very little variation in the responses, in the benefits in our study. But obviously, it is very, very difficult to establish whole diets like this then in an aging population. And this is really where we could think perhaps about some, as dangerous as it is, to look for magic bullets, but also for alternative therapeutic approaches to target this. And the one thing with symbiotic health we're interested in is bifidobacteria. And the reason is simply they are linked to health, and this is data produced by symbiotic health. And you can see there the dominant two species, this Bifidobacterium aldolescens and Bifidobacterium longum. They're actually quite dominant in a healthy gut, but then decline actually the older. As I mentioned, they do respond to this NEMI diet, but they are also, can be targeted by prebiotic, and they can amplify other fiber effects through cross-feeding. And then there are a lot of, and then there is some really interesting links actually in elderly populations, and there's a lot of variation here amongst global populations. I don't want to simplify this too much, but this is a study here on centenarians in Sardinia, one of these populations that eat quite healthy, and you can see in the centenarians bifidobacterium, and specifically this bifidobacterium adolescens, substantially higher than in just healthy elderly, for example. Yeah. And we have quite a bit of functional work now linking B. adolescens, for example, to the production of GABA. We see here that there is studies that it ameliorates sarcopenia, which is often associated in the elderly. This is our own human trial in adults showing that it does improve barrier function. And then finally, this is a nice paper here showing that in different models, it actually increases health span and lifespan that can be achieved through this bifidobacterium adolescens. And with this, I'm out of time. So what the take-home messages is, I think the microbiome is a plausible contributor to aging. In animal models, we have clear evidence. In humans, it's always hard to establish this. Diet is likely a factor of this. To modulate the microbiome, things of just, for example, calorie restriction is already important. But then we have also diet like the Mediterranean diet or the NEMI diet showing that this can actually influence host microbe interactions that are relevant to aging. Here again, calorie restriction might be one mechanism because it is easier to actually restrict calorie with this, but then we have other healthy components of these diets. It's very difficult to implement this, specifically in the elderly and specifically once they are in care homes, but to improve healthy eating, you should really encourage to eat already healthy earlier in life. And finally, there is scope for microbiome targeted intervention to improve health spans, but we need much more clinical trials, specifically also some that look at microbiome mediations. If you would think of how to do a trial like this and what the most important clinical outcome would be in a trial on healthy aging, this is me and Itzik Mizrahi in Seoul in 2014. I'm probably still not completely recovered from that week, but I can say we were in a nightclub 3 times in just 5 days, dancing all night. There's no way I could still do this, but I would be very keen to see a probiotic who can actually help me with that again. Yeah. So these are all the individuals that actually contributed to our NEMI study, would acknowledge all of them, also acknowledge the Papua New Guinean Institute and the people of Papua New Guinea, the funders. And with this, if you're interested to eat a bit more non-industrial, all the recipes are freely available, and you can download the recipe book from the internet. So thank you for your attention.
Thank you, Jens. That was a great talk. I'm going to allow a few questions. While people are coming to the microphone, I may ask the first one. I was fascinated by the switch from short-chain fatty acid to branched-chain fatty acids with the restored diet. And that brings into the question the role of branched-chain amino acids, because one of the best predictors of the risk for type 2 diabetes is actually the plasma level of branched-chain amino acids. So were those affected by the restored diet, or do you think it's mechanisms that are independent from these branched-chain amino acids, and it's entirely gut microbiome dependent?
So unfortunately, I don't have an answer. Yeah, we didn't access them. They didn't show up in our metabolomics analysis, so we don't actually know to what degree they are affecting in a diet like this. Yeah.
Okay.
So, yeah. But you're right, they are one of the strongest links to actually type 2 diabetes, for example. Yeah.
Thank you.
Yep.
Yeah, amazing talk, really interesting. So I have, in a way, I have 2 questions that are completely separate. One is about what you said at the beginning, this theory that after not being procreative anymore, then the microbiome could kill us. But evolutionary, we're like, I mean, it's not your theory, but evolutionary, I guess, 1,000 years ago, we only kind of managed to get to 40-ish. So, but now we have this industrial diet, which is quite unhealthy, as you showed, and we still get much older. Maybe do you have some context in your.
I mean,
right?
I don't think we necessarily get older because of the industrial diet, you know. So okay, to some degree, because of course it provides security, food security. So we've completely, you know, stopped starvation and things. But once this is, I think the life expectancy is medicine, you know, and treatments and better and prevention of infections. And this is also when you actually look in these non-industrialized population, their life expectancy is shorter. But if you actually once, you know, you are out of the period where you have infant mortality, then often they become actually quite older, you know. So I think that the evidence, then, at least from the observational studies, show that these healthy diets actually expand life expectancy. And I don't know if I read into your question now, but clearly evolutionary, none of this probably played a role, you know, because as you said, you know, throughout human evolution, you know, we probably, you know, didn't get that old. But then the hypothesis is still, you know, that there might be a pleiotropic effect. It's a hypothesis. I like it, but I haven't tested it. I don't know if Ruth ever thought about testing it. So, yeah.
All right, and the other one was about the beefidos and the centenarians. I don't know how deep you dug into that, but are they different than the same species in healthy adults or even in kids?
Well, that's a really good question. I actually wouldn't know that. Yeah, and I also have to say this is one population of centenarians where they are very dominant. There are other, and I don't know if Sean looked into some of, but this is quite confusing because there are even centenarians in China then which are dominated by bacteroides, for example, which we then in Western courts actually link more with detrimental effects. So it's one population, and it's, you know, it's one, we need the human trials, but it's just one potential strategy, you know, by which we perhaps can actually do interventions then. Yeah.
Thanks a lot.
Thank you. Very, very short question.
Yeah, very
short
quick.
answer, please.
Yeah, very beautiful talk. I'm curious about the composition of the diet you use in the trial, and I'm wondering, does that diet contain low animal protein compared to, you know, Western diet, control diet, you know, you use in the trial?
So we mimic this on what, you know, rural Papua New Guineans would eat. So it has animal protein. It's chicken, fish, and pork, because that's what they eat. And it's low levels. Yeah, so it's one small portion a day, but it's not vegetarian. Yeah. So, and it's all in the book, so you can download it. Yeah. So as we have the entire diet plan and recipes, they're all there.
Sounds good. Yeah, thanks.
Thank you.
Thank you.